Thank you again for the kind introduction. I've been working on RPA and RAA since 2020, and actually I'll present to you with an example of my first assay I designed, how I actually do this. And you might remember 2020, the pandemic started and everybody was very excited about this, mostly in a negative way. And we thought maybe we could contribute a rapid test, which is very sensitive and actually easy to develop. So we started out with RPA, and we wanted to establish this with lateral flow dipsticks, which looks the same as all the rapid antigen tests, you know. And yeah, so our goals were to have this very sensitive and accurate assay, which also has an internal positive control. The nice thing is that you can multiplex with RPA and RAA. Also, it should be device-free, so everybody should be able to use it. The idea is that you can drive the amplification reaction with your body heat. Also, it should be easy to use and, of course, low cost, so we want everything in a nice package. So how to actually establish this? As I already mentioned, isothermal amplification. It should work at 37 degrees centigrade or lower. It should use freeze-dried reagents and disposable pipettes so that we can actually make a cheap kit out of this, which should also be easy to use. And development is fast. So the procedure we try to establish it's actually cell lysis from saliva or a gargling sample, then reverse transcription and isothermal amplification, and then finally the detection step. And you might notice that here I specified a freeze-dried RT kit for reverse transcription. At that point in time, the problem was that the only supplier of RPA reagent, which was TwistDX, was unable to provide a kit with a reverse transcriptase in it for several years. So we didn't even start with that. So we started with their standard RPA kits and did the reverse transcription before, just so that you know how we addressed this at that point. And now I will tell you just a little about RPA and RAA. I guess most of you are in some way familiar with this, so I will not go into the deep details. When we started, actually there was a personal collection. My boss suggested to use RPA. My boss is Professor Chong, and he actually did his PhD thesis with Olaf Piepenburg in the same building. So the guy who invented the RPA. Okay, so I did some literature work, and if you look at this kind of ugly table, you might appreciate that the RPA is about in the middle, and I marked it for you. And the nice thing is that it works at low temperature, it's quite fast, and it's available as a kit with freeze-dried proteins. And here an amplification time of 30 to 90 minutes is specified. Actually, this is not true. All my assays run at maximum 15 minutes, and as low as 8 minutes of amplification time is fully sufficient. So yeah, we were quite positive about this method. And how does it work really? You don't have to care so much how it works, but you have to know some details to design the assays. And basically, it's a PCR work-alike. And since it's isothermal, you cannot easily melt the double-stranded DNA template you have at least at some point. So how to go about this? Olaf Piepenburg had the idea that you can use proteins from different organisms and some reagents to help this. And this recombinase system basically wedges in primers into a double-stranded DNA template, and then the elongation is carried out by a strand displacing polymerase, to keep it simple. And then you get different, then you get exponential amplification. But what you also should keep in mind, this works at rather low temperature. So the specificity is not as easy to achieve as in PCR, where you can just change annealing temperature. This is here not possible. So we have to use other tricks. And the most important trick is to use a probe. And so a third oligonucleotide. And I will just explain this for one of the possible probes you may use. And this is an NFO probe. This is for lateral flow dipstick detection. And this is based on the idea that you can bind this third oligonucleotide between the primer binding sites to confer specificity. So let's say your primers bind at some random places in the DNA sample you provide, then you might get amplification products which are not what you like. They could even be based on whatever contaminations of human DNA are in there, and you want to detect a pathogen. So the specificity comes from this third oligonucleotide. And this is rather complicated and also a little expensive, but it's the only expensive oligonucleotide in the whole procedure. And the idea is that at the 5' end, you have a label, which is usually fluorescent. And what's very important is at the 3' end, there's no free hydroxyl group. So this probe cannot be elongated when it's when it's not cleaved. So it's a cleavable probe. It can only be cleaved in our system when it's bound to the complementary strand and a special nuclease is present. And this is called NFO. It's E. coli endonuclease 4, and for short, it's NFO. And this nuclease will cleave DNA strands when hybridized to a complementary strand, and there's an abasic site. So if there is no base, it's just the sugar phosphate backbone here. Okay. This is the principle. Then how does it really work? You have your amplicon from the RPA or RAA, which is defined by the forward and reverse primers. And the reverse primer also carries a label. This might be digoxygenin or biotin, whatever your dipstick can take. And then if you have this long amplicon, the probe will bind basically between the primer binding sites. And in this state, it is cleaved, and then it can be elongated. And this will result in a dual labeled amplicon, which has fluorescence at the where the probe contributed it, and at the where the reverse primer contributed a label, there's usually digoxygenin or biotin or whatever. And this allows for specific detection on the dipstick. So the dipstick will provide a binding site for one of the labels, and the other label will be detected by an antibody bound by a gold particle, which will give the red coloration we are all familiar with. Okay, we bought some dipsticks at that point in time from Millennia, which they have a nice explanation how it works. You have different zones. You have zone which just detects any antibody as a positive control, and this dipstick, which we bought, has two different detection zones for specific analytes. And the idea was that we detect SARS-CoV-2 and also an internal positive control, so a transcript from human, to show that everything worked, that reverse transcription worked, and that the amplification worked. Okay, and what you will always get is the upper band. This will be stained, and you might get staining of these lower bands if the analyte is present. Okay, so far so good. As I mentioned, there are different label combinations you may use, which you can decide for your assay. And this is another overview slide which sums it all up. You amplify, If you have probe binding and cleavage, you get this dual-labeled amplicon, which is then detected on the dipstick. Okay, so and now to get to the point, how to design the primers. And I was quite experienced in PCR primer design, but when I started with RPA, there are some complications you need to pay attention to. Actually, TwistDX has a nice assay design manual, and I think information is also in the papers from Olaf Piepenburg. And one very important point is that the primers are rather long. They should be 30 to 35 bases long. This provides a sufficient binding surface for the recombinase proteins, so this makes it easier for the primers to be wedged into the double strand. Shorter primers do not work as nicely. I tried this actually, and you need more amplification time, just as they say. You might use longer primers, but I would not advise for this if you can help it. I will explain this later more in detail, but one big problem is secondary structure formation and cross-hybridization in this assay, which will lead or may lead to false positive outcome. Okay. Other things to consider for the primers are GC content and also the product length. Actually, just as in quantitative PCR, shorter products are usually better. For once, amplification is faster since we do not cycle, so every amplicon that is formed can be used as a template almost immediately. And longer templates are actually worse. Also, regarding specificity, the shorter products always win in the competition for nucleotides and polymerases, as you may know. Okay. Given all that, I used Primer 3 for primer design. Many of you probably have heard of this. It's a very old school software which is available as a web interface. I still like it a lot since it gives many, many possible primers and you have very many possibilities to filter, such as GC content and other parameters. And yeah, I recommend to get into this or use the software you like. For Primer 3, I like that it gives so many different primer pairs that there's always something to choose from. Okay. And I mentioned before that cross-hybridization is a big problem in with these assays. To kind of combat that, I used MFE Primer. I used this as a command line tool, but it's also available online with the web interface. And if this tool tells you that those primers will hybridize, this may be bad. So let's say your probe and your reverse primer hybridize, then you have the fluorescent and the 3' reverse primer label already together, and then everything will be positive on the dipstick. So you don't want that. So I kicked out all the combinations that hybridize with each other, especially with the probe. And then I recommend, I will mention this later, I even recommend to test without amplification for cross-hybridization to rule out oligo combinations which are bad. Okay, and now for the reverse transcription part, it's rather easy. As I mentioned, when we did this assay, we could not get RT-RPA kits. However, from Kitian, and there are now RT-RAA kits, which is a technical workalike. They are available and available also from Xpedite. The reverse primer you already have, this is sufficient for cDNA generation since this will also be used by the reverse transcriptase. And so the good news is no further action is required to make your assay RT ready. You may want to give some more time for reverse transcription and amplification if using this approach. Okay. Then the probe, I already explained that you don't, that you do need the probe for specificity. Of course, only the specific amplicon should harbor the probe sequence. You might want to check if your probe has other possible binding sites in the target genome. And the twist manual says the probe should be 30 bases, so just like an RPA primer, then the abasic site, and then another 15 bases. And what's important is that the 3' part, which cannot be elongated since there's no hydroxyl group, this should be quite short, so that it's not used as a primer after cleavage, so that the recombinase proteins will not spend their time with this trying to wedge it into double strands where it fits. So this should be short. I already mentioned the label, the 3' block, and the abasic site, and it will be cleaved by NFO if everything works as intended. And what I did then, I spent most of my thoughts on the probe, actually, since it's very expensive compared to the other oligos, and also during the pandemic I had to wait for many weeks for it, so I didn't feel like buying stupid probes. So I thought it's probably best to let it bind to a sequence that should not change in your pathogen or other organism of interest where you want to amplify a sequence. And the capital letters I show here are codons for residues which are important for SARS-CoV-2 functions. So this is where it binds to the cellular receptor ACE2. These codons are, I would not call them conserved, but I was hoping they would not change over time. And actually, I put the probe cleavage site just after 2 of these important codons, which I was hoping would stay constant. For other targets, for the positive control, we intended, of course, you should also choose regions which are not, do not harbor SNPs or other sequence variations which might complicate things. Okay, then I got all this, but I didn't stop there. What I also bought is a synthetic double-stranded fragment which represents the target sequence. Why that? It's another 50 euros spent, but it's quite useful. So you can spike it into samples to see whether sample composition or buffer composition impairs the reaction. So let's say your lysis buffer is bad for RPA. You would see this if you use this synthetic template alone and together with this lysis buffer to see if it's harmful. You may also want to detect nuclease activity or other problems in the sample. And it's also very useful to determine your sensitivity threshold. Okay. Given all that, I want to emphasize that this testing of oligonucleotides is very important, even more so than with PCR. So I spent a lot more time. Predominantly to counter these unwanted hybridizations, which are in my hands were a problem. I already mentioned if the probe and the reverse primer are brought together and they stick together, this is bad since it will lead to band on the dipstick, which is false positive. This became even more important after I changed my dipstick system. Previously, I used the Millennia dipsticks, which are totally fine, but I switched to the Kitian dipsticks available from Xpedite. They're much more sensitive in my hands, which is nice, but also you see these unwanted hybridizations actually, you will see them more. And what I did then is I combined the oligos needed for amplification. Actually, all the combinations I was intending to test, I put them separately. I added the running buffer of the dipsticks, and then I just ran the dipstick without prior amplification, so without RPA or RAA reagents, just to see whether this will result in a signal which shouldn't exist if it should just originate from these oligos sticking together. And then all the remaining combinations are used for amplification. with a synthetic template and of course also with real samples. And I prefer then to run also in agarose gels to see what's going on before using the dipsticks on these amplification reactions. Okay, finally, now I will show some data. This is a rather unfinished presentation of these dipsticks. These are the Kitian dipsticks, and I tested different primer combinations here for cross-hybridization without amplification, as I mentioned. And what I would like is an outcome where you can only see this upper band stain and no lower band. On the third dipstick from the left for AE2, you see I have this unhappy smiley because there's a very prominent lower band, which tells me that this is this does not work. This is false positive. Other combinations were nicer, but I actually showed this because I had so many bad combinations, which is probably due to the target sequence, which was one of the more complicated assays I designed. And with the non-false positive combinations, of course, I can continue. But now let's switch back to the spike assay I promised I would show you. And this is an agarose gel. I will walk you through it. I have different samples. Sample 1 is a negative cDNA, negative because it's not the cells were not infected with SARS-CoV-2. Sample 10 is a sample from infected cells, and I also have the S sample, which is the synthetic spike DNA template representing my target sequence. And on the left, I have two markers. Then I have the two cDNA samples without reverse transcription. This is an important control. All the bands which appear here should be unspecific since they're not from cDNA, but just from the DNA present in the sample. And you might appreciate that for the spike assays, you only see here some diffuse staining. But for the RPL27, there is a band also without reverse transcription. This is a different target. This is for internal positive control. Then with reverse transcription, with some imagination, you see a signal coming up in the lane number Roman 4, but not with the uninfected cDNA in lane 3. And then I did a trick. I incorporated with this reverse transcription kit, which was, it's not the RT-RAA kit I intended to use initially, but an extra kit. As I explained to you, RT-RAA was not available at that time. I added the reverse primer for reverse transcription, and this boosts the band we are getting. And we are also getting this band in the lane number 7, just with the synthetic template. And I was quite happy about this. This seemed to be specific, judging from the agarose gel. A negative control in lane 8 is negative. For the RPL27, this was not as nice since we also get the band without reverse transcription, so I would rule out this primer pair. Okay, and then for the final assay, I show you a less busy slide here. On the left, there's a negative control, and then I put different amounts of the synthetic spike template, 5 picograms and 5 femtograms, and also from mock infected cells, a cDNA sample, and from infected cells. And yeah, I guess it works in some way. You may also notice here that in the negative control on the left, there's some very weak staining. And I can explain this to you later. This is probably carryover contamination from aerosols. So basically, it's a PCR we are running. And if we don't work free of contamination, then there will be some carryover. So just as a caveat. Okay. To sum it up, what do I recommend? Design the primers and probe according to the guidelines from Olaf Piepenburg. They work quite nicely in my hands, and I changed some parameters, and I could see that, yeah, probably he's already spent some work to to make this work nicely, and changing things is sometimes not a good idea. The tools I use are Primer 3 and MFE Primer. My recommendation is to design more primers than for PCR, since you want to kick out more combinations probably. Test for cross-hybridization without template. I would also recommend to use the synthetic template for different purposes. I might add that I ordered all oligos from IDT. I'm not affiliated with IDT. I'm just a customer. In my hands, they always worked. Probably there are other suppliers who can do this. I also recommend the agarose gel and be generous with amplification time. So I usually ran this for 15 or 20 minutes in the beginning. All remaining combinations which might be specific, test them with the dipsticks and optimize the reaction time. So if your signal is nice, just go down. No need to over amplify. You might want to determine the sensitivity threshold, so how many molecules you need to get a signal. And you may want to identify possible challenges with this double-stranded template. I recommend the Kitian reagents. They're readily available from Germany and within Europe. They are sensitive. Pay attention to carryover contamination, and then I hope you will get your results within minutes. And actually, this is my final slide. I don't have too many conclusions. RPA for S detection, also for the spike coding sequence from SARS-CoV-2, works on cDNA samples. Carryover contamination is an issue. Multiplexing, which I cannot show today due to time reasons, this is possible. You can detect different amplicons in parallel. It's a bit harder to establish since more oligonucleotides may cross-hybridize, and they may also interfere with each other during amplification, but it's possible to find combinations. Yeah, and as I mentioned, testing. Not too many people were involved in this project. Most of the work was actually done by me, so surprisingly. I got the samples for SARS-CoV-2 from our Virology Institute, and I have a long-standing collaboration with Megacore for other relevant templates. And also I have very nice interactions about these assays, so we are still learning how to do this even better. Thank you very much for your attention. Are there any questions?
Thank you very much, Till. We may have had some technical issues with some of the attendants that could not see the slides, but anyway, I just want to reassure everyone, we will send you the replay, and it will be online in the next couple of days, and we will be very happy also to take separate calls, or I'm sure Till will also be very happy to hear from you if you have any any questions, or if you are interested in the collaboration as well.
Yes, sure. Thank you.
Till, there is a short question here.
Yes.
Could RPA primers be tested in PCR or couldn't? I mean, I often use PCR for initial screening of LAMP primers, and it works excellent even with long FIP and BIP primers. Do you think this makes sense to test your primers with a PCR system first?
Yeah, actually you could, but to be honest, I was too lazy to do this. I just went for the RPA since it's also so rapid. I felt this was more convenient, and yeah, actually I'm a fan of testing with the same system you intend to use in the end. So I don't see a point against this, but yeah, actually I wanted to save time. I hope this answers your question.
Yes, thanks. And Tatsuo Serikawa is asking, is a forward primer required, or only the probe and reverse primer are enough for the reaction?
In theory, you could go with a probe, but then you would lose the specificity thing. So if you use just a forward primer with fluorescent and a labeled reverse primer in combination, then you will detect all the amplicons which are possible with these two primers. If you have this probe, which is dependent on like a third sequence, then you can kick out all the amplicons which are not what you want. And I think it's a big issue. So the approach is basically the same you use for TechMan assays, if some of you might remember. This was, it's still being used, but you use them if you cannot get a specific PCR, or if there are other reasons to use it, of course. But if you cannot get a specific PCR because the primer binding sites are too similar between different targets, then you use this third sequence in the middle. And I would strongly recommend to do this, since on this agarose gel I showed there were not too many bands, but there are actually other amplicons, and I would be very much afraid that it's not specific. As I said, you cannot control annealing temperature in this assay. It's all the same temperature, and this temperature is quite low. So yeah, I would recommend against it. Thanks.
Christian is asking, all the enzymes you use for RPA, RAA are commercially available from Kitian?
Mm-hmm.
Yes.
Everything is from Kitian. As I mentioned before, you can also buy them from Twist, but the last time we bought from them, there were customs issues. They're now shipping from the US. Kitian worked actually better. The dipsticks are very nice, and also the amplification kits work better in our hands even.
Yeah, to that we hold stock here in Munich. So we are the exclusive distributor for Kitian in Europe, and we always have stock, usually enough stock for all your requests. So don't hesitate if you have questions about RAA, RPA to contact Xpedite's team. We have a lot of experts. Actually, Ejona is going to speak in a minute. And we have stock there in Germany. Maria Rita de Felice is asking if we have a denaturation step before starting the amplification, or if it's not necessary. So it's not necessary.
Yeah, not necessary.
That the whole reaction takes place around 37 to 40 degrees. Igor Oskorbin is saying, thank you a lot. I just found that PCR can be easier to run if testing of many primers combination is necessary. So I guess you mentioned multiplexing. Yet it has different buffer condition, etc. So would magnesium and DNTP concentration have to be optimized, or do you need to optimize those concentrations? You don't,
Yeah, I think I never did this. So for DNTPs, you cannot do anything. This is fixed in the kit. You can optimize magnesium. I think it just doesn't have to be limiting. So in my hands, this was not essential. And I changed the primer concentrations, and this didn't work out. So the concentrations given by Olaf Piepenburg in my hands, they were quite good already.
Yeah. Yes, that's usually also, we get a lot of requests for researchers willing also to fine-tune or to tweak and to optimize or try to optimize the different enzymes, and if they can have the reagents separately. And I have to remember everyone that this job has been done. When you buy a Kitian kit or you buy a RPA kit, you have optimized reagents, and the companies have put a lot of efforts into optimizing it and making them robust as well. So it may be possible to do everything, even an epsilon faster or whatever, but at the end, the reagents have been optimized to be robust, to be working in the optimal way, using those really premixed. That's why actually none of the reagents are separate. They're all pre-lyophilized in the tube as well. Yes. Did you also try lower temperatures? I don't know, Teal, that's an interesting one. Have you tried lower temperature ones?
Yes, I hope you can still see the overview. I actually did this. I hope I have it in here. No, sorry. I don't have the slide in here. I went down to 28 degrees, and it works a little slower, but it works. And this is 37, but different minutes. So you see that this signal comes after 8 minutes. But
Yeah.
Yeah, sorry, I don't have the lower temperatures, but I actually tried it extensively in my hand, and I think Sigrun tried it like in the armpit, and this works reproducibly if you have a nice assay, so you don't even have to mind the exact temperature. Of course, when I do it in the lab, then 37 degrees, it's fastest and best, but lower is possible.
Yes, I actually remember a researcher telling me that he was doing the amplification under his armpit for a few minutes.
Yeah. This is actually Sigrun's trick. She said Ziploc bag and just put all the samples in your armpit.
You can do that. So actually, it's very interesting question because RAA enzymes will start to work at room temperature. That's also why when you prepare your reaction and you add the magnesium, you add your sample and you close your tubes, the reaction is starting. So you should not wait too long before you put them into if you're using fluorescence or into your incubator or whatever your system you're using, because the reaction starts already. So that's a very, one could see it as a bit of a limiting factor. It's actually a great parameter when you want to do point of care systems, because you don't need to have heating capacity to higher temperature than basically 37 to 40 degrees. No need for betaine, DMSO, or such additives. No, exactly. Everything.
No.
Everything is there. Well, thanks you very much, everyone, for all your questions. Thanks a lot, Teal, for your great presentation. We will make the material available online on YouTube to replay this webinar. But now I would love to transfer the microphone to Ejona. Ejona, do you want to take over and present a little bit of what you... Yeah, perfect. Thank you.
Okay. So hello, everyone. As was mentioned already, I'm going to talk a little bit about our journey in developing a duplex RAA assay for schistosoma detection. But first, I wanted to give you some background onto schistosomiasis and why we decided in the first place to undertake this assay development. Schistosomiasis is, sorry, let me just activate my laser pointer to make it easier to see. Yes, so schistosomiasis is a parasitic infectious disease that has a significant global burden of disease, as you can see from the map of affected patients here. It impacts the health of more than 200 million people worldwide, especially in the countries with lower to middle income, in the tropic and subtropical areas, which is why it has been classified as per the WHO definition as a neglected tropical disease. It is caused primarily in humans by 3 major species, which you can see down here. Schistosoma japonicum is more prevalent in Southeastern Asia, while the other 2 species, Mansonae and Hematobium, are co-endemic and prevalent in African countries and the Middle East. These 2 are also, Schistosoma mansonae and Hematobium are also the strains that we are targeting with the duplex assay that we have developed. In the diagram here on the right side of the slide, you can see the what I would call the vicious cycle of transmission of the parasites. The worms lay eggs in our bodies, which we then excrement with feces or urine, and this way the eggs get transmitted into bodies of water, where they then hatch and infect their next host, the environmental host, which is snails, where they further develop and then end up reinfecting humans, and then the cycle goes on and on and on, which is why I called it a vicious cycle. There are a few diagnostic tools already used at the moment on the field in schistosomiasis research. The first one is microscopy. Very easy and straightforward. Using this method, you can visualize and count eggs of schistosoma parasites. The drawback of this method is that in cases of low parasitemia, so when the infection is not at a very high grade, or where there is an infection, but in the stool sample, let's say there are no eggs visible, the infection would go undetected. So it's not a method that has a very high sensitivity, and this can be an issue in diagnosing correctly. Another method is a lateral flow test that detects specific antigen, the CCA, that in urine that is a biomarker for schistosoma infection. The issue with this is that, unfortunately, it is not very specific, and it has been shown that it has a pretty significant rate of false positive results, which again can cause some issues when trying to do proper diagnostic of a population. Finally, in order to combat the low specificity of the CCA test and the low sensitivity of the microscopy, we move on to qPCR, which is, as I'm sure all of you know, the reigning king of molecular diagnostics for the moment. It can be, depending also on the assay design, it can be super sensitive and very specific to the species and the strain that you want to detect, but it has a major drawback compared to the first two methods that I just described, and that is the requirement for infrastructure. This led to then the rise of a need to find a method that can do what PCR does in terms of analytical performance, so be very highly sensitive and very species-specific in schistosoma diagnostics, but also have the field-friendly component that the other two methods have. And here is where RAA enters the game, and isothermal nucleic acid amplification techniques in general. So one of the major drawbacks of PCR when we're talking about field friendliness is the fact that it works through thermal cycling, as we all know. It cycles constantly between, I would say, moderately and high temperatures, and this uses up a lot of energy. So the need for power and electricity is quite high. And this is the main reason behind the requirement for a solid and proper infrastructure. With RAA, the whole amplification, as Till also mentioned, is in his example, happens at, I would say, low to moderate temperatures, so around 37 degrees, and sometimes as high as in the lower 40s. And this already reduces the requirement for energy a lot, since there is no cycling and there is no need to heat at such high temperatures. This already reduces the energy needed, and also it is much faster than PCR is. So we can get a reliable results nowadays in 15 to 30 minutes for most assays. And both of these combined together lower the need for a very strong and solid electricity network, which make it a lot more field friendly. We know that for PCR, we need this thermocycler in terms of devices, and most of the thermocyclers that are in the market right now are not exactly very portable. However, for RAA, what you would need is just a heat block, which in cases of amplifications like the one that Till talked about so far, that is very close to body temperature, you would be the heater. But in case this temperature of amplifications go a little bit higher, then you would need a small portable heater or a water bath and a readout platform. This can be a lateral flow test like the one that Till showed, or it can be a device that measures fluorescence while the amplification is going on. Then another aspect that is very, very important when we talk about point of care and field deployability is the amount of material you need to take with you to the field and the amount of material that you need to dispose of. Because most of the RAA reagents come in freeze-dried form already in pre-filled tubes, you don't need to take as much equipment and as much consumables with you to the field, and also the amount of waste that is generated is much lower. And all of these together make it possible that the RAA basically builds a bridge between the high analytical performance of laboratory-based methods and the practicality that is needed at the point of care in the field. But why did we decide to go for a duplex assay? Why not stick to 2 singleplex assays that detect each of the strains that we're interested in, so Schistosoma, Hematobium, and Mansoni? Why build a duplex assay? There are benefits in both the fields of environmental and clinical testing to the simultaneous detection of these 2 major human schistosome species. When it comes to clinical testing, in areas where these 2 strains are co-endemic and coexist in the same environment together, a duplex assay would help to avoid misdiagnosis and misclassification, and therefore improve knowledge on how to treat the patient much faster and much more efficiently. You would basically just need to run 1 test and know if it's either or of the strains. And this would greatly support control and elimination programs that are running on these endemic areas. And in terms of environmental testing, you would be able to monitor transmission sources such as, for example, water samples or snail DNA extracts, and these would help you map risk areas more precisely. And also, recently there have been hybrids of Schistosoma mansoni and Hematobium species, and a duplex assay such as the one that we designed would help monitor these hybrids and their occurrences. So we started the development process. We wanted this to be a quick development, so we didn't spend a lot of time in designing our own singleplex assays, but we turned to literature and tried to find and source singleplex assays from there. For Schistosoma hematobium, our first strain, we found this paper that described an RAA assay that targeted the COX-1 gene of the Schistosoma hematobium species. They claimed the sensitivity of down to 10 copies per microliter within 10 minutes of amplification, and also a high specificity. What was interesting to us is that they had already tested for cross-amplification against Schistosoma mansoni, which is the other half of our duplex assay, and there was no cross-amplification, so the assay was still highly specific even against this strain. Similarly, our source Schistosoma mansoni assay from literature was also tested against Schistosoma hematobium, that's the gray SH that you see in the right graph here, and was found again to be highly specific. For Schistosoma mansoni specifically, this assay that we sourced from the literature targeted the mitochondrial minisatellite regions of the Schistosoma mansoni genome, and they claimed a super high sensitivity of down to 1 copy. If you look at the reddish brown graph here, down to 1 copy of the target within 20 minutes of amplification. All good so far. In paper, these seemed perfect assays to combine into, or a great start, at least to start the journey towards a duplex assay. We decided to test these in our lab and in our hands to see if they can perform similarly. And what we found was that for Schistosoma hematobium, the LOD was, so the limit of detection was around 170 copies, and it took 15 minutes to positivity. And for Schistosoma mansoni, the picture was even more different than what we saw in the paper. Anything under 1900 copies, so 1900 copies, was undetectable, even when extending the amplification time up to 30 minutes. So then we thought, okay, let's get to optimizing these assays as a duplex to improve their performance and make them work as best as possible in this duplex reaction environment. And we see if maybe by tweaking these different assay parameters, we will be able to improve the sensitivity of the assays. The first parameter to consider when optimizing the assay was temperature. In the individual papers, the schistosoma hematobium assay was the temperature of amplification was 39 degrees, and for Mansona it was reported to be 42 degrees. So we tried a range between these two temperatures to find the temperature at which both of these assays would perform reliably well. We tried a bunch of temperatures and we landed on 40.5 as the temperature of amplification for the duplex assay. When it came to the primer and probe concentrations, the standard ones used normally are 420 nanomolar of the primers, or of each of the primers, and 120 nanomolar of the probe. Both of the individual assays we got from literature used the same concentrations, and we also tried this in-house, and we saw, for lack of a better term, an overwhelming of the reaction because of the high amount of primers and probes that was in there in the duplex reaction. So we tried then to optimize it a little bit, optimize these ratios a little bit. We tried different things. We tried keeping one of the assays with the primer probe concentrations at the standard 420-120 ratio, and then halving the other one. We tried the opposite. We tried having both of the primer probe concentrations for both of the assays. We then tried an asymmetric approach where the concentration of one of the primers was different from the other primer. And out of all of these configurations, we landed on keeping the standard 420 nanomolar primers and 120 nanomolar probe for the schistosoma mansoni assay, and then halving the concentrations for the hematobium assay. The reported time for amplification was 10 minutes in the paper where we found the schistosoma hematobium assay, the singleplex, and 20 minutes for Mansoni. But seeing as we had a reduced sensitivity in our hands than what was reported in the papers, we decided to increase the time of amplification up to 30 minutes. And then again, because it's a duplex assay and you need two different signals at two different channels, then we had the ROX probe for the schistosoma hematobium and a FAM fluorophore for schistosoma mansoni. However, after doing all of this work and optimization and different configurations of different parameters, we still found no improvement on the sensitivity of the schistosoma mansoni assay. So then we went back to the drawing board and we thought, okay, what we are struggling with is just the schistosoma mansoni assay. And we thought of assay that is very widely used and has been validated for a long time and has been used in diagnostics for a very long time, but it is a PCR assay. It's the SM127 assay that targets this highly repeated randomly arranged DNA sequence in the schistosoma mansoni genome, and it is very highly repeated. It comprises almost 12% of the genome of this strain. And we thought, okay, because it's so highly repeated, it's meant to have very high sensitivity, and it has worked very well as a PCR assay. So why not try to convert that into an RAA assay? And that is what we did. And to do that, we based our work, our design work, on the design principles of RAA that if you scan the QR code here, you will be linked to. They're also published on our website. But I'm going to go through them a little bit. I'm not going to go into a lot of detail because they match with a lot of what Till already shared. So the amplicon length is suggested to be between 80 and 200 base pairs. It's not preferred to have a higher length of amplicon because the higher the length of amplicon, then the more it takes for the amplification to occur. And the point of RAA and of a point of care assay is to be fast and be rapid. The primer length is supposed to be between, or suggested to be between 30 and 35 nucleotides, with a GC content varying between 30 and 70%. What is important is that you try to avoid having long stretches of guanine in the 5' end, because that has a negative effect on the amplification. And for the probe, there are a few more things that you need to take into account. It needs still to be a considerable length to have a proper strength of binding to the strand, to the target strand. It's suggested to be between 46 to 52 nucleotides, and it needs to have a blocker modification on the 3' end to avoid extension, so that it's not extended as a primer would be. This could be a C3 spacer, an amino moiety, or a phosphate group. It needs to have a fluorophore and a quencher, and between the fluorophore and the quencher, there need to be 2 to 4 nucleotides spacing. And one of these bases in between the quencher and the fluorophore needs to be an abasic nucleotide that can then be cleaved to generate the signal and release the fluorophore from the quencher. And another parameter you need to take into account is that the fluorophore needs to be attached at least 30 nucleotides distance from the 5' end of the target sequence. Yes, so using those, we created our SM127 RAA assay now from the PCR assay, and we tested it, and we found with very much relief that we now had the specificity and sensitivity that we wanted. You can see that for hematobium, we still have the results that I showed you before, around 170 copies detectable within 10 minutes of amplification time. And for schistosoma mansoni, from the previous assay where we had 2,000 copies limit of detection, we can now go down to 120 copies within 10 minutes of detection. So now we finally could say that we have a rapid, specific, and very sensitive tool for the simultaneous detection of schistosoma mansoni and hematobium. It is very easily field deployable as an isothermal amplification method. And the final parameters for the assay are 10 to 15 minutes of amplification at 41 degrees Celsius or 40.5, depending on if it's the device where you're doing the heating allows you to do a 0.5 temperature. It's a fan-ROX multiplex, so it needs signal to be generated and recognized on a green and orange channel. And then the standard primer probe concentrations, as I mentioned, were kept for schistosoma mansoni, even with a new assay, and the halved primer probe concentrations were kept for schistosoma hematobium, so as to not to overwhelm the duplex reaction environment. I then wanted to make sure that the new SM127 assay is also very specific against schistosoma hematobium, and there wouldn't be any cross amplification. So I set up the duplex schisto assay with the primers, the reagents, and everything, and then spiked in 18,000 to 180,000 copies per each of the reactions of the schistosoma hematobium positive control. And I would expect increasing signal in the ROX channel, which would be down here, and it's the graphs you see down here as expected. But there is no positive signal, no unspecific signal detected in the FAM channel, which is the upper 3 graphs that you see here, which was good news. It meant that the SM127 assay is specific and does not cross-amplify the hematobium strain, which meant that we had a successful, theoretically, duplex assay. And then the next step, logically, would be to test it with biological samples. And I would say one of the most difficult samples when it comes to schistosomiasis is snail DNA extracts, because it is known to have a lot of inhibitors and compounds in it that affect the performance of the assays, be that PCR, be that isothermal amplification. There's a lot of inhibitors being co-extracted with the DNA, unfortunately. So we had access to 4 snails that were confirmed with an infection of Schistosoma mansoni. We had DNA extracted from them, and then we had the result benchmarked against the qPCR. So the DNA extracts we ran with the Schistosoma mansoni qPCR and with the duplex assay. And as I try to color code the CTs for each extract to the RAA graph, so you can see we had the CT values ranging from 12 up to 27, and they correlate perfectly to the time to positivity, or basically the lower the CT, the earlier the time, the increase in signal in the RAA. Another very difficult sample type when it comes to schistosomiasis research is cfDNA extracts from plasma. So you have an infected patient with Schistosoma mansoni in our case, and then you extract cell-free DNA from the plasma of this patient, and some of this cell-free DNA is also going to be DNA that has been released from the Schistosoma mansoni cells. So you try to detect these using, in our case, the duplex assay that we had just developed, and this time we benchmarked against microscopy. So for these samples, we had the results of a Kato-Katz test, which is a method of basically counting eggs per gram of stool of the patient under microscopy. And again, I try to correlate the colors of each of the extracts to the graphs that we saw for RAA. And again, we see amplification, we see positive signal quite fast. The latest is around 4 minutes, 4 to 5 minutes. And just to make it easier to see this correlation, I plotted this data into the graph from the 2 different sample types that I showed you. So here on the left, you can see in the bar chart, you can see the time to positivity that it took with a duplex RAA assay to detect the Schistosoma mansoni DNA from snail extracts. And on the right side, with the bluish dotted line, you can see the qPCR CT value, and you see that it correlates quite well. On the right side, you can see, again, with a bar chart, the time to positivity with a duplex RAA assay, and this time in blue, the counted eggs per gram of stool with a microscopy method. And here still there is some correlation. So we see basically we see positivity throughout. Again, it's not as perfect of a correlation as it is with the qPCR, because in the case of qPCR, it's basically 2 different methods of detecting the DNA load, the pathogen DNA load in a sample. So these are easy to compare. In the case where you're comparing a molecular method to a microscopy egg count, again, there is the correlation is not as high in terms of sensitivity, of course, but in terms of positivity, we were able to detect all of the samples in quite low times to positivity. And then as a last step, if I look back to my graphs that I've shown you before, you always see this dip at around 3 minutes. And this is because it's always recommended that for RAA assays, as Stephen and Till mentioned, as soon as all of those reagents come in contact with each other and with the sample, the reaction starts immediately, because it is doable at very low temperatures. So it starts immediately, and unless you mix it after some time, which is suggested to be between 3 to 5 minutes, you might have a non-homogeneous reaction mixture, and you might have depletion of reagents in one part of the reaction mixture and overload in another. So it is recommended to do this mixture. However, what I wanted to test here was automatic mixing, which is doable with the Axin reader, which is the device that we use here in-house. The sensor that does the reading of the fluorescence in each of the tubes basically travels from tube to tube. And when it does, it also has a magnet attached to it. And if you insert a steel ball into your reaction mixture into your tube, then every time this sensor will move, it will move the steel ball with it. So it basically does an automatic mixing while detecting the fluorescence in each of the tubes. And what I wanted to see is how this improves sensitivity of the assay. And in each of the graphs, you can see with the uninterrupted line, the same conditions as with the dashed line, but the uninterrupted line is with this auto mixing with a steel ball, and the dashed line has no mixing. And you can see that the auto mixing leads to a much earlier rise to positivity compared to the no mixing method for Schistosoma mannsoni on the left graph. And it shows positivity down to 120 copies, while for the no mixing method, only 2,400 copies are visible, but lower than that, we're not able to detect anything anymore within the 10 minutes of amplification. For hematobium, it looks a little bit more different. So for hematobium, when I tested 18,000 copies, the auto mixing and the no mixing didn't have much of a difference with such a high input into the reaction, but as the more you reduce it and the closer you get to the LOD, the more you see the difference. So with 1,800 copies for auto mixing, you can see here the rise to positivity is much earlier than with no mixing. And for 180 copies, which is the line that just starts rising here, for auto mixing, you can see already a rise to positivity, even if it is later on, but for no mixing, it's not there. And I apologize for the graph because I just noticed that the legend does not match the graph. I don't know what happened there, but I hope you understood my point. And then just to wrap up, and to kind of show you how involved and how invested we are in field deployable testing, especially for point of care testing and how RAA fits into that. I wanted to show you the Xpedite toolbox, as I like to call it, or the ecosystem of things that we have created to support this field deployable testing. Our job already starts in the lab, so you can already, if you're planning to do some point of care or field testing, you can already contact us before you leave for field testing, and we can already help you with our assay design, either as a contractual service where we can do it for you, or give you our consultation services to help you design a proper RAA or RPA assay. And then when it comes to the field, we start with the extraction of nucleic acids, where we have a wide array of kits and products for extraction of nucleic acid from a variety of samples, then down to isothermal amplification reagents, which are from Kitian, as Stephen mentioned, steel beads for auto mixing, or the Axin reader, which is the device that I mentioned that does the amplification, so as a small heat block, plus the readout of the fluorescence signal. And then further on, you can combine all of these into an equipped mobile laboratory that has everything you need for point of care testing, including the Axin reader, so the amplification device, which you can see here. Yeah, that is it. Thank you for listening.
Thank you so much, Ejona. Thank you very much. It's a great presentation and a lot of work.